This is a production of Cornell University.
Okay, so one, I want to thank all of the industry members who have been sponsoring our expo. I'd like to thank Gemma, who's a backbone of this part of the symposium, who is keeping us all together and doing the work behind the scenes. I'd also like to thank all of Cornell Cooperative Extension personnel who have worked very hard to pivot this meeting from an in-person event to a virtual presentation. And last but certainly not least, I'd like to thank Dr. Thierry, who is my co-presenter for today. He is an assistant professor at Rutgers University. He is an excellent weed scientist working in specialty crops. And he is a phenomenal collaborator, not only from extension outreach opportunities such as this, but also research trials that we have together. And I am extremely lucky to have the opportunity to be working with him. So what I'm going to do is I'm going to get on to our introduction to herbicides. And this is going to be the first talk that's going to be the background for the remaining of the topics today. And this specific presentation, obviously, we're going to have to talk about what the problems are with weeds. This is our kind of obligatory slides of why you should care about this material. We'll talk about herbicides and the history of herbicides and herbicide use. We'll talk about why herbicides don't work, and then we'll talk about herbicide sites of action. Specifically how we're going to classify them for the details that you're really here for, the sites of action. So again, problems with weeds, they are direct competitors with crops that result in yield loss, and that's competing for water, light, and nutrients, but they also reduce harvest efficiency. They can actually stop men and machinery from moving in the field. Species like dodder or mistletoe or broomrake can parasitize the crops themselves. They can be the host for pests or pathogens of crops. They are dangerous or noxic, poisonous, toxic, hazardous in their own right. Think poison ivy or giant hogweed. And they disrupt ecosystem services and aesthetics. So where I came from most recently out in the Western US, Medusa head alters fire cycles, and that has huge implications for many ecosystems in an entire region of the US. Now, if we go back to yield losses, which tend to be very important, what we think about primarily is weeds, if they are not managed, can result in substantial yield losses. So this is work that has been done by the Weed Science Society of America, the Yield Loss Committee. If we have best management practices in place, but we don't manage our weeds, they estimated that in our major crops, we could see anywhere from 50 to 71% yield loss. And that can account for hundreds of millions of dollars a year in economic losses, up to billions of dollars a year in economic losses. So we need to manage weeds. And the primary tool that we're using for suppressing this unwanted vegetation are herbicides. Now, herbicides have been the dominant pesticide applied in our major agronomic production environments, so corn, soybeans, cotton, across time. But that doesn't mean that they aren't important or aren't significant or aren't highly used in specialty crops as well. So the figures on the left-hand part of this slide, that green bar at the top of each figure for corn, soybeans, and cotton, that's herbicide use. And that's historical herbicide use going back to the 50s and 60s. And you can see how heavily we relied on these chemicals, particularly in our soybean systems and in our corn systems. But again, like I said, that doesn't mean that we aren't using herbicides extensively in our specialty crops. On the right-hand side of the slide, these figures are for some select specialty crops: apples, blueberries, peaches, onions, pumpkins, and snap beans. maybe we don't have quite the same degree of separation in the amount of herbicides we use relative to insecticides or fungicides, but we're still applying herbicides to significant acres across the United States. So if we just want to think about historical chemical weed control, we've been using herbicides for quite some time to manage unwanted vegetation. Now, they might not look quite the same as the products that we're used to using today. So for instance, in the 1800s, there's documented use of lime for control of equisetum species. In 1902, sodium arsenite for the control of water hyacinth. One of my favorites, 1915, the report of the amount of salt that they were using for the control of field bindweed. This was, at the 3 to 5 ton per acre limit along railroads to suppress this vegetation. But what we're really kind of interested in, what we're going to focus in, is our synthetic herbicides and the start of the chemical era of agriculture. These products began in about 1942. So this is a timeline of the release of herbicide sites of action, all the way from the early 1900s to the most recent site of action that was released, the group 28s. What I've done on this slide is I've highlighted all the different WSSA groups that you might be familiar with. So the group 4s, the group 5s, the group 6s. So 4 would be auxins. 5 and 6 are our photosystem 2 inhibitors. 14 are our PPO inhibitors. The 27s are our HPPD inhibitors. Now you can see is that there was an era where we had a lot of herbicide discovery and release between the 50s and the 80s. And then we have kind of a dry spell until 28 was brought out, and that was just brought out. So many of our herbicide classes are what you might consider old. And the big question is when are we going to get new ones? Everyone wants to know when is the new herbicide mode of action coming out? And I'm going to tell you, we shouldn't necessarily be relying on this, again, this kind of exponential growth of novel herbicide mode of action released. The group 28 just came out. FMC announced this. There's a new herbicide active ingredient. It's part of a new class of herbicides, but don't get too excited. It's primarily for use in rice production systems, which aren't dominant here in New York. And then the next announcement that I have seen that even suggesting about a novel, herbicide mode of action, site of action, is from Bayer, and saying that they anticipate it coming out in 2030. So this is going to kind of be the hallmark of some of the stuff that Thierry and I are going to say, at the base of what we want you to, why we want you to understand our herbicides is we need to preserve these chemistries over the long term because we aren't going to be getting new technologies. Now, what are some of the constraints in place that might be preventing the release of new herbicide sites of action or new herbicide active ingredients? Part of this is the cost of discovery and commercialization. Part of it is it's a biological constraint. It's the availability of target sites within plant cells that herbicides can go after, so to speak. And then there's the consolidation of chemical companies, which may influence herbicide discovery. So in case you're wondering about the cost of new herbicide release, this is from Steve Duke's paper in 2008. He estimated that it costs $246 million to bring a new synthetic pesticide to market. And there are additional costs on top of that for re-registration of products, for adding new active ingredients, changing use patterns, registrations in different states. New York may have additional regulatory hurdles that chemical companies need to address, which can cost money to be able to bring a new pesticide to market here in the state specifically. And then there's the biological constraint. How many target sites are there within plant cells that herbicides can go after? So understand that herbicides bind to specific sites within the cell. And these target sites have to be critical for plant survival. So you've got to find a site that you can't have an alternate pathway around. It's got to be a lethal inhibition. You don't want to have a comparable target site in a non-target organism, particularly as mammals, for safety reasons. And then a target site, you don't want it in great abundance because you want to be able to easily inhibit this target site with low use rates. With respect to chemical company consolidation and the impacts on competition and commercialization, I've seen it argued both ways. So it suggests that the evolution of our chemical companies and their consolidation leads to less competition. Now we're talking about these bigger companies involved in discovery and development. That's not to say that there aren't a lot of smaller companies out there that are engaged in this process, because there are. But we tend to think about the Bayers, the ChemChina, the FMC, the Cortevas, the BASF. So are these consolidations resulting in less competition? But there are other people who are arguing, because these companies are also consolidating their seed company profiles, that this could actually facilitate innovation because it may encourage them to maximize trait technology and therefore to investigate new pesticides. We're going to talk about herbicides. We obviously want to talk about herbicide failure. And herbicides are not always effective. We use them, but they're not going to be 100% effective. And there's a lot of reasons why we can see herbicide failures. Sometimes it's we pick the wrong herbicide to go against a target weed, or we don't use it at the right rate or spray volume. We're not using the suggested recommended additives, such as adjuvants. We may have antagonism. We may tank mix two products, and the use of both of them at the same time may reduce the efficacy of one or other, or there could be spray solution issues, such as water being too acidic or not acidic enough, that can affect the performance of these chemicals. The weeds might just be too tall at the time of application and we don't get complete coverage, or they might be stressed. Stressed weeds, particularly with our systemic herbicides, don't respond well to the chemistries that we're applying. There could be herbicide or environmental factors affecting either the deposition or the activation of these products, and then there's herbicide resistance. And herbicide resistance is an evolutionary process where we have a species, a population where we have this transition from susceptible to resistant due to genetic changes that are already present in the population that limit the efficacy of the herbicide. And what happens is that our repeated use of certain chemistries over time and space removes all of the susceptible plants and leaves the resistant ones present, and they just continue to reproduce, survive, and thrive in these systems where the selection pressure is in their favor. You can think of it this way. We used to be able to kill this weed with this herbicide at this rate, and now we can't, okay? But I'm gonna let you know the situation is pretty significant. Right now, worldwide, we have 509 unique cases of herbicide resistance. We have herbicide resistance that's been found in 266 species around the world to 164 different herbicides. And the United States leads the way with 123 cases. Now, Weeds can be resistant to more than a single herbicide at a time. It's not that there's a lot of populations out there that are resistant to just Roundup or maybe just first-rate herbicide or this herbicide or that herbicide, but there are populations out there that are resistant to multiple herbicides at the same time, meaning that using both of these herbicides, you can't control it with one and you can't control it with the other. Sometimes the resistance, there's one mechanism that's controlling the resistance for different herbicides. And sometimes we have multiple mechanisms, and we differentiate those from cross-resistance and multiple resistance. And with multiple resistance, meaning more than one mechanism that enables plants to survive treatment with herbicides that differ with respect to their sites of action. And I'm going to use this figure. This is some herbicide resistance screening we've been doing in New York in horseweed. So higher bars mean more dead plants. So population 7, 100% death from both Weathermax and first-rate herbicide. So this population is susceptible to both of these herbicides. Population 9, we have 100% death when we used Roundup Weathermax. However, we have very little death in this population when we use first-rate herbicide. So it's resistant to one herbicide, one site of action, because Weathermax and first-rate have different sites of action. Population 17, we didn't control it with Roundup. We didn't control it with first-rate. So it is resistant to both of those chemistries. Now, this is actually a significant concern when we're talking about populations with resistance to multiple sites of action, to multiple chemistries, because as we start to accumulate these multiple resistances in our weed populations, it reduces the size of our chemical toolbox that we have available for management. This is some data that Dr. Ian Heap has been collecting. It can be found at the International Survey of Herbicide-Resistant Weeds, weedscience.org. I definitely suggest everybody go have a look at this site. It's quite eye-opening to look at our herbicide-resistant issues here in the United States and around the world. Of those 509 cases, he's found and identified 60 populations that have resistance to 2 sites of action. There's 21 populations out there, weed populations out there, that are resistant to 3 sites of action. So 3 different herbicides, active ingredients. 13 are resistant to 4, 5 are resistant to 6, and we have 1 that's resistant, or 6 are resistant to 5, and we have 1 that's resistant to 6. If I'm correct, and I'd have to look it up, I think there's a ryegrass population in Australia that might be resistant to 7 or more herbicide sites of action. So it's a significant problem. And again, if we start accumulating resistances, multiple resistances in a single population, we start reducing the efficacy of the products that are available to us for managing unwanted vegetation. So if you're curious about whether you have resistance or not in weeds that you're not well able to control, there are some questions that you can ask yourself. First and foremost is asking if you've done everything right. Were you using the correct rate? Were you using the right herbicide? For instance, stinger, clopyralid, it's effective against a very limited number of species. It's good against the aster family and the legume family, but it's not going to provide control of other broadleaf plant families. So if you're going after a weed that isn't in those 2 target groups, you might not see effective control. It's not resistance. It's just that product never was going to work well in the first place. So are you doing everything right? Are you doing everything according to the label recommendations? And are you applying it at the correct time? Are you using it against weeds that are at their most sensitive stage? And let's face it, that's going to be small and succulent and not stressed. Were there any environmental conditions that could have affected your herbicide performance? Did you put out a post-emergence herbicide and then get rain immediately after that could have washed the product off the leaves? Or did you put out a residual soil applied herbicide and then you didn't get a rainfall event that was needed to incorporate that herbicide into the soil solution? Are there patterns in your field that could suggest maybe something went wrong with your spray program? So for instance, when I was out in California, I got called out for, I think we've got an issue with resistance. And every eighth row had weeds. That's not herbicide resistance, that's a clogged nozzle. Okay, so that's a pattern suggestive of something else that's not related to a genetic evolutionary change leading to resistance. But if you have live plants that are intermixed with dead plants of the same species or other susceptible species, and all these plants were at the same size when you treated them. So if you treated at a 2-leaf stage, a 4-leaf stage, according to the label recommendations, and everything was done right, and you killed some, but not others, then we should start to be concerned that we're looking at a case of herbicide resistance. Or if you have a problem that's getting worse over years, you have a patch that seems to be growing, or a weed that seems to be expanding in your fields when you're using a same herbicide or same class of herbicide over time, then we want to be concerned. And or if you know that you have cases of herbicide resistance nearby, maybe for instance with horseweed, and horseweed has seed that can be dispersed on wind currents, and a neighbor has a problem with this, and now you have a problem with this, then we should be thinking about whether you have herbicide resistance. And if you do have herbicide resistance or you suspect, you can reach out to your CCE personnel. You can reach out to me and we can collect seed and we can do screening because we definitely need to know if we have novel cases of resistance developing in our state. So if you do suspect that you have herbicide resistance, or if we want to mitigate or prevent the development of herbicide resistance, the best thing you can do is understand the biology of the weeds that you're going to be dealing with in your field. The better that you understand the biology, the more able you are to build a diversified, integrated, and dynamic program to suppress those unwanted individuals. Make sure you're starting clean and you are staying clean as possible. So planting weed-free seed, maintaining your field as clean as possible, and preventing field-to-field spread. That includes managing field edges. Again, for instance, something like horseweed, it's dispersed via wind currents, and letting it go in one site can facilitate problems developing in a remote site. Managing weeds at harvest and post-harvest to prevent the buildup of your seed bank. Using multiple herbicide modes of action at labeled rates, again, recommended weed sides, and diversifying your chemical strategies both within and between seasons. So we're not enhancing or increasing a selection pressure for one specific product. Scouting your fields regularly to identify any escapes early. And, understanding that we don't have to rely solely on chemistries for managing unwanted vegetation. So adopting cultural and physical strategies as you can to manage weeds in your fields, on your field edges. So now we're going to, this is just our introduction to our next talk, which is going to start looking at the herbicide sites of action. Is when we're talking about herbicides, there's a lot of different ways that we can describe or that we can classify them. And we use a lot of these words when Thierry and I give these talks, we throw out this terminology, and then sometimes we combine this terminology together. There, these classifications are not independent or exclusive. So we can talk about controlling weeds about the spectrum of control, whether we have broad spectrum, we get a lot of species, or very narrow. So selective versus non-selective. The movement within the plant. We're going to talk about herbicides that are contact products. So they burn the tissue that they come into contact with, and therefore you need good coverage. Or we're going to talk about products that are systemic and translocated in the plant. So they're applied and then they are moved within the plant to where their target site is. We talk about timing. We'll use timing relative to weeds, so prior to their emergence or post-emergence, but we also talk about timing relative to the crop, so pre-plant, in-crop, or post-harvest. Then we talk about modes of action and sites of action, and that's what's going to focus here today. And I'm going to tell you that this isn't always easy to understand because sometimes we use these terms interchangeably, and I'll show you an example of this next. So mode of action is defined as kind of like a big overall picture, the general way in which a herbicide controls susceptible plants. Whereas the site of action is talking about the very specific biochemical site and process that's disrupted. Again, sometimes mode of action and site of action are used interchangeably. And I'm gonna use this as an example. So the left part of this slide is a screen capture I got from the University of Wisconsin. They break up one of their handbooks, modes of action, amino acid synthesis inhibitors. And then they talk about the specific site of action, the WSSA group 2 as being the ALS inhibitors, the ALS inhibiting herbicides. Whereas the HRAC website, if you look at the right side, talks about that exact same group 2, the ALS inhibitors, and calls it the mode of action. So understand that different groups may have different words for describing the same thing, but Thierry and I will talk to you what we mean by the site of action, which is going to be the specific biochemical pathway. So for instance, this is just an example, bromoxynil. It's the mode of action, it's a photosynthesis 2 inhibitor. It generates reactive oxygen species. Its site of action is specifically, it binds to the D1 protein at histidine 215. Okay, that's a difference between mode of action and site of action, general versus specific. So this is how we're going to break up today's talks. Thierry, this is the HRAC mode of action classification poster. You can get this at hrac.com. I think it does a nice job about grouping herbicides into those herbicides that generate light activation of reactive oxygen species. And then those herbicides that affect cellular metabolism and those herbicides that affect cell division and growth. Down here in this red box, this is going to be our sites of action. This is what Thierry and I are going to focus on. But specifically, we're going to break it up where Thierry's going to talk about the reactive oxygen species generating herbicides. That's going to be our next half hour. Then I'm going to talk about the herbicides that disrupt cell metabolism, division, and growth. And then Thierry's going to come back and talk about keeping those products on site, on target, and preventing environmental concerns and injury to our crops and off-target organisms. So again, we're going to break it up this way. Thierry's going to do ROSs. I'm going to do the other two categories, just so you can see what's coming. The reactive oxygen species, those burners, those are products like Besancon, Callisto, and Goaltender, Rely, and Command. The cell metabolism, division, and growth inhibitors, products like 2,4-D, Dicamba, and Allion, Matrix, Roundup. So thank you very much. I'm going to stop sharing my screen and I am going to pass it off to Thierry and he can take our next topic.
Thanks, Lynn. I will start sharing my screen. hopefully it will work nicely. Can you see my screen?
Yes. You can see your screen, but you might need to take it into presentation mode.
Here we go.
Excellent.
Okay. All right. So, well, thank you, Lynn, for inviting me today to the New York Expo for presenting together on herbicide and to give this lecture on the 101 basically of herbicide, the basics of our weed science. Thank you, Steve, also for inviting me today. Well, yes, I agree, Lynn. When we're working together, we're really, I think, a great team of weed scientists, and we are doing a lot of research together. So it's really something that I enjoy doing a lot. But my talk today was really, it's really following what Lynn explained regarding herbicide site of action. And we'll be really focusing right now on the photosynthesis inhibitors. Before I start, really, if you have questions, don't hesitate to put them in the chat box. Sorry for the French accent, but it's already too late for me to change it, so I will keep it. But if you have any question, please do not hesitate to put that in the chat box. Lynn explained it, but really, it's really important to understand the difference between mode of action and site of action. So eventually, because they're very used interchangeably by weed scientists, and it should not be the right way to do it. We should be a little bit more precise when we're using this term. So mode of action is really the biochemical mechanism by which an herbicide will cause growth to cease in a targeted plant. If you're looking at the site of action, that's the exact location. That's exactly where the herbicide is acting in the plant. It can be an enzyme, it can be a different type of molecule in the plant, but that's where the herbicide is really acting. The mode of action is a consequence of, of this binding to something in the plant. So for example, if you're looking at the allosulfuron herbicide, Sandea, this one, Sandea will inhibit acetolactase synthase, which is an enzyme in the plant, which is involved in the synthesis of branched chain amino acids. So by binding to this enzyme, allosulfuron will shut down the production of branched chain amino acid in the plant. And that's what will cause a plant to die, because these amino acids are essential for plant growth. If the plant is not able to produce them anymore, the plant will die relatively quickly. So herbicides are classified by site of action. That's the way we are classifying these days. So the classification code is required on the label. So for most of the products that you will find in the US, you will have the code really on the first page on the label. I give you an example, gramoxone, paraquat. You look on the top right corner, you see group 22 herbicide. That's indicative of what kind of site of action this herbicide is targeting. So that's targeting the photosystem 1 of the plant. So that's an herbicide that we'll be discussing a little bit later in the presentation today. Lynn discussed herbicide resistance, and really what we're doing today, it's important with regard to herbicide resistance management. Most importantly, one of the very important recommendations for delaying or preventing the onset of herbicide resistance is really to rotate herbicide site of action and eventually use a mixture of different herbicide site of action for preventing the selection of a specific weed biotype that may be resistant to a specific herbicide. So knowing these numbers is really important, because that will help you to effectively rotate your site of action when you are applying some herbicides. So Lynn presented this poster, which is really classifying all the herbicides that are available on the market these days. I will really focus on the photosynthesis herbicides, the ones that are targeting the photosynthesis. Just to give you an example, on this slide, you look at the green box on the top left. These herbicides are known to inhibit photosystem 2, which is, there's two different types of photosystem in the plant where the photosynthesis is taking place. There's photosystem 1, photosystem 2. We have some herbicides that are targeting photosystem 2. Actually, you have two numbers. You have number 5 and you have number 6. Why? Because you have the same mode of action, but you have two different sites of action. You have herbicides that will target a specific protein, and some herbicides that will be binding to another location on this protein. So they will be classified in two different sites of action. So looking at photosynthesis, that's really, that's really the basic of the plant. Without photosynthesis, there would be no plants. On that, we have a variety of herbicides that are really targeting photosynthesis. So just as a reminder, photosynthesis, it's when the plant are taking up carbon dioxide from the air to produce sugars via photosynthesis. As a result of this photosynthesis process, oxygen is produced as a product of photosynthesis, and the plant will also produce glucose that will be sugars that will be used in other process, in the plant. And the plant really needs the light energy. The plant are pretty much only one in the living world to use light energy for really realizing this photosynthesis process. To look a little bit more into the detail, and it's important to understand what's going on right here, because that's where some of the photosynthesis herbicide will be targeting. So basically, the plant is absorbing photon, absorbing light, and that will generate a chain of electron transport. So basically, the plant is absorbing the photons, the energy from the sun. It's breaking down water molecule that will generate electrons that will be transported through different molecules. And in the process of transport, that will generate energy for the plant. So the plant is not using directly electron for making other process, but the electrons that are transported are helping to the generation of energy in the form of ATP, adenosine triphosphate, or NADPH. And this molecule of energy, if you want, will be used by the plant for making other products, for making amino acids, for making sugars, for making lipids, for generating everything that the plant needs. But really the basic is right here. It's a production of energy that the plant will need later for other chemical processes. On herbicide, we'll be targeting some molecules that are really important in this photosynthesis process. So the first group is really the WSSA group 5 and group 6. This one are inhibiting photosynthesis at photosystem 2. And actually, if you remember what I presented in the previous slide, these herbicides are binding to the D1 protein, during the electron chain transport. And by binding to this protein, they are blocking the transport of electron in the photosystem. And by doing that, I mean, if the plant is still collecting energy from the sun, but if you don't have transport of electron within the cell, that will generate a lot of highly reactive molecules that we are calling reactive oxygen species. And these molecules will be reacting with pretty much everything which is forming the plant. So especially lipids. Lipids and proteins that are really important for the membrane of the cell, of the plant cells, will be attacked by reactive oxygen species and will be degraded relatively quickly. You have different group of herbicide in this photosystem 2 inhibitors. You have the triazine, such as atrazine and simazine. You have the triazinone, which is a different chemical family with metribuzine. You have ureas, diuron, and linuron. 2 herbicides very important for veggie production. In the nitrile family, we have bentazone, which is also a photosystem 2 inhibitor. So these herbicides, trazine, trazinone, ureas are very often, most often soil applied, eventually early post-emergence. So relatively quickly after the weeds start emerging, but you don't want to wait the weed to be too tall to apply these herbicide. But they are really mostly considered as residual herbicide. They are absorbed by both the shoots and the roots, and they are only translocated in the xylem of the plant. So really the xylem is where the water is moving in the plant. So that's where these herbicides are moving into the plant. The nitrile, bentazone are early post-emergence herbicide. They are mostly contact herbicide because they are not translocated in the plant. They are not moving from the roots to the shoots. They are really acting where they've been sprayed, where they've been eating the plant. Some soil-applied photosystem 2 herbicides, such as atrazine or simazine, can carry over to following sensitive crop. And I took a picture, I borrowed a picture to Dr. Alan York at NC State. showing some symptoms of atrazine carryover on tobacco. That's what we'll be discussing in the next few slides. We'll be discussing what is the symptomology that is characteristic of this photosystem 2 inhibitor herbicides. So really, I mean, this photosystem 2 inhibitor really is what is really outstanding with them. What you will see very frequently is really leaf chlorosis. So yellowing of the leaf turning pale, and after a while, it will start generating necrosis as well. So you will have a lot of intervenal chlorosis. So the main veins of the leaf will stay green, but everything between the veins can start to turn yellow. You will have some marginal necrosis or curling on the margin of the leaf. And what is really typical with this photosystem 2 inhibitor when you're looking at injury, the oldest leaves are always the ones showing symptoms first. And if in my previous slide on tobacco, you could see that the basal leaf of the plant, the two first leaves of the plant were the ones showing symptoms. Actually, the new shoots were still looking good, looking green, but really the oldest leaves are showing the first one to show symptoms. You can see that again on this picture. This one is a picture of trazine injury, polyatrazine on cucurbits. And again, I mean, it's a nice picture to really visualize how these herbicides are causing damage. You see a lot of yellowing on necrosis later on between the veins of the leaf. The veins are still green. And if you're looking at the picture on the left, you see that the new shoots, I mean, the newest leaf, the smallest leaf in the center of the plant are green, but the oldest leaf have been the one showing injury from photosystem 2 inhibitor herbicide. A few other slides showing some symptomology, but this time with metribuzin. Again, the older leaves will show intervenal and marginal chlorosis, followed by dieback. You have some curling on the margin of the leaf. In some situation, you can look at the gray picture on the bottom right. You may have some mottled or intervenal chlorosis that will develop after necrotic tissues have dropped. Especially in bushes such as blueberry, if you apply this herbicide on new blueberry cane that have not been hardening, that are still green, that don't have a solid bark, you may have necrosis of young tender stem exposed to the spray on this one. And with this herbicide, you will not see root injury. Lynn will discuss some other type of herbicide that will be causing a root injury. With this type of herbicide, when you have damage, it's really affecting the foliage of the plant, not much the roots of the plant. This one is terbacil. It's a very specific herbicide that we're using in blueberry, which I'm familiar with because I'm working a lot with blueberry. We are really careful not using it a lot because it's really a dangerous herbicide if you want, potentially causing injury in sandy soil. But again, you will see the same kind of pattern with pale green to yellow leaves turning brown on dying. You may have brown or red spots. Again, marginal and intervenal chlorosis is typical of terbacil injury as well. I mentioned blueberries, but yes, definitely here in New Jersey, we're growing blueberries in sandy soil. In this type of soil where you really, it's a very permeable soil, in some situation, you don't have always a lot of organic matter, so you don't have any buffering capacity of the soil if you want. And we know that terbacil can be extremely injurious to blueberry in some situation, depending on the type of soil. Another type of herbicide affecting the photosystem is glutamine synthetase, and it's really is only pretty much one herbicide in this group that is widely used. It's glufosinate, Rely. So this herbicide is really targeting the glutamine synthetase enzyme activity, and that's the enzyme that will convert glutamate to ammonia onto glutamine. And that's something which is really important for amino acid production in the plant. When you're blocking this enzyme, what will happen is that you will have a lot of ammonia accumulation in the plant, and this ammonia will destroy the cells and directly inhibit photosystem 1 and photosystem 2 reaction. In addition, the high ammonia, because it will change the pH of the cells, the pH gradient across the membrane, and by doing that, you will prevent basically the production of energy by the plant because you need to get some gradient pH between inside the cell and outside of the cell. If you don't have this gradient, you don't have this exchange for the cell, you don't have this energy production, and that will prevent the plant to grow and to develop. So some characteristic of glufosinate, do not confuse glufosinate with glyphosate. Okay, we are really talking about a different herbicide than glyphosate. Glyphosate is not a photosystem inhibitor herbicide, if you want. So glufosinate, it's quickly absorbed to soil. It's very rapidly degraded. It has absolutely no soil activity. It's strictly a post-emergence herbicide, so you need to apply it on emerged weeds. It has limited translocation, meaning that it will not just stay where you apply it on the leaf. It may move a little bit around, but it will not move through the xylem or to the phloem of the plant. It has some really limited translocation. In most of the cases, it will act much like a contact herbicide like paraquat. It's a good herbicide for controlling many annual broadleaf weeds. It's a decent herbicide for annual grasses when they are relatively small, on small seedlings. Exception is goosegrass. Goosegrass is not really well controlled by glufosinate. Anything outside of this type of weeds, anything such as nutsedge, perennial broadleaf weeds, or Bermuda grass, which is a perennial grass, will not be efficiently controlled by glufosinate. So that's a limitation of this herbicide. What is the symptomology of glufosinate? So it's really killed at the site of droplets. You have slight translocation, but not to the entire plant, something completely different from what you would expect from glyphosate. So you will start to get some initial burning within a few hours after exposure. So it's starting to show symptoms relatively quickly. If you are comparing that to what we just discussed before, the photosystem 2 inhibitors such as atrazine, metribuzin, herbicide, it will take days before you starting to see some injury. With glufosinate, yes, within a couple of hours, you can start to see some physical effect on the plant. So after this initial burning, basically after, I would say that most of the time after a week, the plant will really die after a week. So injury symptoms include leaf burning, leaf distortion, leaf crinkling, and necrosis. It's relatively quick, compared to, if you were to compare to glyphosate that Lynn will discuss later, glufosinate is killing the weed more rapidly than glyphosate. A few things that are really important to keep in mind with glyphosate if you use with glufosinate. Sorry, if you use glufosinate, the rate of injury or the efficacy of the herbicide will be increased under bright sunlight condition. Usually, I recommend the growers to start using glufosinate. Rely, I would say, between 10 a.m. and 5 p.m. We have done experimentation when I was at NC State looking at applying glufosinate at very early in the morning. And it's really contrasting. If you really applied it when there is no light, you will drastically reduce the efficacy of the herbicide for controlling weeds. It's also a good idea to apply glufosinate under high humidity condition, and you have a good plant and soil water balance. Again, that's the idea that this herbicide need to be applied when the weeds are under good growth condition. If the weeds are stressed by drought or by temperature, these weeds may not absorb the herbicide. The herbicide will not be as efficient for controlling some of these plants, some of these weeds. So there is crops that have been genetically engineered for getting resistant to glufosinate by introducing putting the PAT or the BAR genes to this plant. So basically what these genes are doing into the plant is that they will detoxify glufosinate by attaching an acetyl group, if you want, to the glufosinate molecule. And by doing that, it makes the glufosinate completely inactive. So it's not toxic anymore to the plant. So right now, there is only 3 crops that have been developed for being resistant to glufosinate. We have corn, soybean, and cotton. Yeah, I mean, I show a picture as well of some injury of glufosinate in high-bush blueberry. On that's really new canes, new shoot at the base of the plant, and you can already see after 7 days, you can already see the necrosis that glufosinate may be causing to these new shoots. So again, a few pictures. So you can see that on blueberry, it can be tricky on blueberry because you can cause a necrosis of the growing point on these new canes. So they are relatively small. If you don't have a shield for protecting new canes from herbicide spray when you're applying glufosinate, you may get this kind of injury. And if you're killing the tip of these new canes, basically the new canes will not keep growing. And that's really a problem because in blueberry, we are regenerating the plant by producing new canes. If you don't have any new canes, that will be an issue. You have some drift injury from glufosinate applied between rows on summer squash and cantaloupe. On the left picture, the plant did not recover. On the right picture, maybe because it was a little bit higher on plastic, we got drift, we got some leaf severely damaged, but the plant managed to keep growing. That may still affect, the potential yield of the plant later on. The PPO inhibitor, so this one, group number 14, WTC group number 14, these one are targeting the protoporphyrogen oxidase enzyme, which is very important enzyme that help producing a molecule which is a precursor molecule for chlorophyll on heme, which is chlorophyll is really needed for photosynthesis, for harvesting solar energy. The heme is needed for electron transfer, and I show you why electron transfer were important in photosynthesis. So these herbicide, by targeting an enzyme which is really important in this process, will really cause injury. I mean, prevent the plant to do photosynthesis. So in this scenario, in this case, when you are inhibiting the PPO enzyme, that will also cause production of reactive oxygen species that again will attack and destroy lipids on protein membranes. So that includes some herbicides that are frequently used in specialty crops. You may be familiar with sulfentrazone, carfentrazone, oxyfluorphen, fomesafen, flumixazine. All of them are PPO inhibitor herbicides. So for most of them, they really have limited translocations. They will behave as contact herbicide. They are typically applied pre-emergence for most of them. There is one carfentrazone, which is post-emergence because it has only foliar activity. So we are only using it as a post-emergence herbicide. So one of the big concerns in specialty crop with this herbicide is that the soil persistence. So there are residual herbicide applied to the soil. The soil persistence will vary depending on crop species. So with some of them, depending on the rate, depending on the soil, depending on the kind of crop that you're planting the next year, there is a risk of carryover with some PPO herbicide. Fomesafen, flumixazine, and oxyfluorphen have both soil and foliar activity. Carfentrazone, I mentioned it, it's strictly a foliar herbicide, strictly applied on emerged weeds. They are primarily used for controlling broadleaf weeds, but it's worth noting that sulfentrazone can also be applied for providing sedge control. We don't have many herbicides that are controlling sedges. On sulfentrazone is one of them that can provide some decent activity on sedges. I was mentioning before the risk of carryover, and I just extract this slide from fomesafen label and look at some, for some of the crop, you have to wait up to 12 months. And if the crop are not listed right here, you have to wait up to 18 months before planting back some of these crops. So the risk of carryover with this PPO herbicide, it's not something to take lightly. You really have to read the label to make sure that you will not put yourself in a situation where, okay, I applied the herbicide this year, but if I plan to plant this crop next year, make sure that you read the label for making sure that you will not be in a situation where you can have potential carryover injuries. Another group of, still some PPO herbicide injury. Most of the time, the injury will look like water-soaked spot on the leaf. These spots will turn brown, and just within a few days, the complete plant, the entire plant will die. And it's really killing only at the site of droplets. And I will show you that on the next slide. That's a slide of blueberry damage that I observed a few years ago in New Jersey. And actually, it's not drift damage. It's not coming from spraying the herbicide. Actually, the herbicide was applied on the ground as a residual herbicide pre-emergence. But I told you before, we have sandy soil in New Jersey. And the grower came a few days later to apply a fertilizer, drove the tractor. It was extremely dry soil. It created a lot of dust. The dust went into the atmosphere, went back onto the leaves and created all these injuries because basically the herbicide was attached to dust particle, went back onto the leaves, and you just have to get dew in the morning. That will allow the herbicide to get back in solution, okay, and to be absorbed by the plant and to create this damage. So this kind of what we're calling shotgun effect of PPO herbicide, that's typical of PPO herbicide injury, either as physical drift, but also as dust drift in that case. Inhibition of photosynthesis at PS1, so it's a different area. So before we are looking at photosystem 2, now we are looking at photosystem 1. On this one are really, there's two herbicides, paraquat and diquat. So this one will inhibit photosynthesis by competing for electron transfer in photosystem 1. So basically, when the herbicide is reduced by an electron, so it will scavenge electron during the photosynthesis. This herbicide will rapidly transfer the electron to oxygen, forming highly reactive superoxide. So these reactive oxygen species, and again, you will start the process of degrading, damaging the cell membrane of the plant. So these one are post-emergence contact foliar applied herbicide. They are really good on broadleaf species, not so much on grasses. They have no translocation, they have no residual activity. Actually, when you're applying paraquat, it's relatively rapidly inhibited in the soil because it will bind to soil particle relatively quickly. So it will have absolutely no residual activity. Some symptoms. So the symptoms with this herbicide are showing up very quickly within 2 hours. If you have the right condition, you will see these symptoms. So PS1 inhibitor will be, the activity will be increased when you under bright sunlight, when you have high humidity, when you have a moist soil. This condition again will enhance the activity of this herbicide. So the symptoms are typical. You will have leaf water soaking within hours of treatment, followed by necrosis within 4 to 6 hours. On spotting, I put a picture right here, but it's again on blueberry that was paraquat drift. You may be used to hear about 2,4-D or dicamba drift, but any kind of herbicide can physically drift. And actually, in that situation, that was paraquat drift injury on high bush blueberry at the time of blooming. And actually, the growers, some of these flowers were killed by paraquat, and we didn't get production on this field. So something really to be careful with. Some typical injury again. Mostly necrosis really relatively quickly. You don't see the necrosis that you will see with some other herbicide. It will be mostly necrosis. In that situation on the right side, that was diquat that was spread on strawberry leaf. And actually, because the deep vein on this leaf will act as a drain, basically it's the veins drain the herbicide that was that drifted on this leaf and accumulated the herbicide at the bottom of the leaf. That's why you have higher injury really at the basis of the leaf. Last group that we'll be discussing today, the pigment inhibitors, the bleacher. You see why we're calling them bleacher, because they're turning really weeds completely white. So this one are carotenoid biosynthesis inhibiting herbicide. Basically, what they do is that they are interfering with the production of carotenoids. And carotenoids are extremely important because they are the molecule in the plant that will protect chlorophyll from photooxidation. So if there's too much energy, the chlorophyll molecule is relatively sensitive. The carotenoids will be there to protect chlorophyll and scavenge any excess of solar energy that may reach the plant. So we have different groups because they are acting at different sites, group 12, 13, and 27. This type of herbicide will be really controlling many annual broadleaf species on some grasses. Clomazone or norflorazone are only applied to the soil. Mesotrione, which is in group 27, can be applied to the soil and also post-emergence to the foliage of the plant. So the symptoms are very evident. That's probably the easiest symptoms to decipher when you have some herbicide injury. So it's really the leaves are completely turning white because you don't have protection of the chlorophyll. So chlorophyll is really degraded extremely quickly by light. So the effect on the plant is that if you have clomazone, the old tissue are usually affected first. If you have vapor drift, it may be generally that the newer leaves will be affected first. Mesotrione is more systemic. You can have symptoms on both old and on new leaves. Some picture of herbicide, HPPD herbicide, clomazone carryover on vegetable crops. So you will see a lot of green. In some situations, it can be relatively similar to what you will get from photosystem 2 inhibitor by showing a lot of chlorosis, but you will really have this kind of white flash, not just saying yellow, but turning white. So that's typical of this HPPD inhibitor herbicide. This white is some common injury, probably from vapor drift on eggplant, on tomato. Again, on this kind of crops, I mean, the newest leaves will be the one showing the most injury with this type of drift. And the last one that I wanted to mention too is that these herbicides, for some of them, can be also effective on sedges. I had this picture coming from here at the station where we applied mesotrione on annual sedge, and clearly you can really see the sedge is turning white. The picture on the right is my favorite weed. It's Carolina redwood. That's something that is a problem here in New Jersey in cranberry. And that's another good type of mesotrione, is a good herbicide for controlling mesotrione. Again, I mean, that's really visual. You can see that only the new shoots are turning white, showing that the herbicide is moving into the plant, translocating, moving to the new shoots and force them to turn white. With that said, that was my last slide on Lynn. I think you have a few other type of herbicide to discuss today.
Great, thank you, Thierry. By the way, there is a question. If you could just in the chat to reiterate the pigment inhibitors, just the numbers of the groups.
Yes, so the pigment inhibitor is group 12, 13, and 27. So I think 27 is mesotrione, norflorazone maybe group 12, group 13, clomazone is group 12 as well, but 12, 13, and 27.
All right, so we're going to jump into the next group of products that we're going to talk about. Thierry discussed the herbicides that are associated with the generation of reactive oxygen species, which tend to result in burning of tissue. Ultimately, I'm going to talk about these two categories: cellular metabolism inhibitors and cell division and growth inhibitors. Specifically, I'm going to talk about these categories, the group 2s, which are ALS inhibitors, the group 1s, 15, 29, 9, 3, and 4. So we're not going to cover all the classes because a lot of these other classes don't have a lot of herbicides in them or aren't used readily in our production environment. So we're going to go in numerical order, and we're going to start with WSSA category 1, and this is the inhibition of the acetyl-CoA carboxylase. Oops, sorry. So that first group. So these are going to be herbicides. In the group 1, they're absorbed through the foliage. So foliar applied products, and they're translocated to the growing points where they inhibit meristematic activity. And they specifically cause damage to the plant by inhibiting the ACC enzyme. And the ACC enzyme catalyzes the first step in fatty acid synthesis. And you're going to be like, why is this important? And we're going to have this, why is this important? Why do I care? Where we explain how these herbicides are acting. And it's important that this herbicide acts the way it is because fatty acids make up the tails of lipids, and lipids comprise plant cell membranes. So they're comprising the organelle membranes and the greater cell membrane at large. So if you remember back to your basic biology, on the left-hand side of the slide, this is a lipid. There's the head, and then there's the fatty acid tail. And then on the right-hand side, this is the lipid bilayer, your cell membrane lipid bilayer. And what happens is those yellow tails, that's what the ACCase herbicides prevent the development of. And if we can't make these lipids, we can't develop fully formed organelles, and we can't develop that plant cell membrane, and therefore it affects the ability of the plants to grow and develop if you can't make these basic structures. The ACCase inhibiting herbicides, there's 25 active ingredients in this group, and these are going to be our FOPs and our DIMs. So sethoxydim, fluazepop, panoxidine. These are our grass herbicides. And the symptoms that they produce tend to be yellowing, wilting. Sometimes you can see that reddening and the purpling associated with crop damage. And because these herbicides, they move to the meristematic tissue and they affect plant growth, plant expansion, plant development, you can get necrosis and dead heart occurring, where you have the death of these meristematic tissues. And basically, as you can see with this corn plant, you just pop that head right out because that tissue has died and is not able to develop further. So the second category is going to be the WSSA2 category, and those are going to be the herbicides that inhibit acetolactate synthase. All right, so they're going to be the herbicides in the blue box at the top of this section. It's a very big chemical group and has a lot of important herbicides that we use. So these herbicides, they can be foliar or soil applied and can be absorbed by both roots and foliage, and then are translocated to their sites of action. Now, I'm going to tell you with the ALS inhibiting herbicides, they might not be equally effective against the same species if they're used pre-emergence versus post-emergence. One interesting thing that we found when I was working out in California in our processing tomato production systems is that matrix rimsulfuron, which is in this group, applied post-emergence, didn't do much for suppressing field bindweed. However, if we used it pre-emergence, we were actually able to see some suppression of the perennial field bindweed vines as they were emerging from the ground. So you can have very different weed species that are being controlled, whether you're using it pre-emergence or post-emergence. These herbicides inhibit the production of branched chain amino acids. So specifically the amino acids leucine, isoleucine, and valine. God, why is this important? Amino acids are the building blocks of proteins, and proteins are structural components of plants, and they're also the enzymes that carry out the duties that are encoded by the genes within the plant nucleus. So this is, if you remember, again, back to basic biology, proteins are chains of amino acids strung together. The ALS inhibiting herbicides interfere with the biosynthesis of these three amino acids specifically. So proteins that rely on these amino acids in their code won't be formed properly because you're not going to have these components of the strand. So again, nucleus, we have our genes that leads to the translation of the genetic material into the protein, building that amino acid chain Specifically, the ALS inhibitors interfere with these three enzymes and our ability to build proteins reliant on these enzymes. Now, this is a big chemical class. It's got 57 active ingredients and 7 chemical families. Some of the components, some of the active ingredients you might be familiar with are the active ingredients that are part of Pursuit and Matrix and Raptor and Sandia. So the symptomology of these herbicides, sometimes we see the undersides of leaves purpling. Sometimes we might just see the red or purple color in the main vein and in the side veins of the leaves. We do see wilting associated with this and necrosis and plant death, but also at really, really, really low rates, we can see leaf distortions that sometimes might be confused with the group 4 herbicides, the auxins. So sometimes we think that leaf strapping and leaf deformations are characteristic of that category alone. No, very low doses of ALS inhibiting herbicides and glyphosate as well can sometimes mimic some of those responses. This is a picture of some leaf distortions that you're starting to see on hemp, but I've also seen it on tomatoes out in California, where we had a non-registered ALS inhibitor herbicide drift onto the tomatoes at very low rates and cause very unusual and unique leaf shapes. The third category that we're going to talk about is the WSSA group 3, and these are going to be herbicides that interfere with microtubule assembly. So this is the category in the blue box up under cell division and growth, a very small chemical group, but an important one and one you're probably all familiar with. So the microtubule inhibitors are soil applied, and they're primarily for the pre-emergence control of annual grasses and some broadleaf species. We do tend to think of these as grass herbicides predominantly. They're absorbed by roots and shoots of emerging seedlings, but not readily translocated within the plant. And they bind to tubulin proteins, so proteins at the growing end of microtubules. Why is it important? Microtubule expansion is inhibited, but microtubules are incredibly important because they are the plant's cytoskeleton. They keep organelles within the cells in place, and they also form the mitotic spindles that are needed for cell division. And if you interfere with microtubules, you are going to interfere with cell division and plant growth. So this is my crude figure of a microtubule. What happens is that microtubules, there are 2 proteins, the alpha and the beta tubulin, they bind together, and then they bind to the microtubule that is growing. The microtubule inhibitors interfere with that growing step. So you don't have these microtubules that get bigger or longer, but you also start to have the back end of the microtubules being broken down. So you're destroying the cytoskeleton of the plant. Here's a better diagram of the plant cell. You can see where the red arrow is, the cytoskeleton, the microtubules. You can see they're kind of drawn visual role as part of the cytoskeleton. Again, remember back to your biology classes where we have the mitotic spindles and we have our chromosome separation and associate it with cell division. And that's how our plants get bigger, by having cells dividing, and we're interfering with that process and the ability to grow and organize. So this is got 20 active ingredients, 3 chemical families. They include the yellow herbicides, treflan, pendimethalin, aryzalin, ethylfluralin. You'll know these can be familiar and frequently used products. Symptomology of the microtubule inhibitors, you can get stem swelling and stubby roots. Again, you get them taken up by the roots and shoots of emerging seedlings. And then you have that inhibition of cell division and root development. So you get those stubby roots. You can see some stunted growth and sclerosis. You can see leaf puckering and other deformations, but really kind of characteristic hallmark of this is the stubby roots, the swollen stems. Last year, we did a study with hemp, and we put down some pendimethalin in some of our trial sites to look at crop safety, and then it rained a lot, and all of the stems were swollen, and we spent a good part of our summer. Is this due to the rain, or is this due to the pendimethalin, or is this a combined effect? The group 4 herbicides are our synthetic auxins. This is going to be our chemistries that you're very familiar with, such as our 2,4-D, our dicamba, our clopyrrolid. These are products that are readily absorbed through roots and foliage and translocated to meristematic tissue. Okay, so they don't just affect what they contact, they're moved within the plant. And they don't just disrupt a single growth process, they disrupt multiple ones. In fact, we don't even know how the auxins are fully working in plants, because they can affect protein synthesis, they can affect nucleic acid metabolism, they can stimulate ethylene production, which can have its own impacts in the development of these plants. It's important because really what we know them for is their impacts on cell wall plasticity, because they're affecting the cell wall plasticity. That's what leads to a lot of the distortions in the plant structures that further impact growth and development that we're really used to seeing. So that's going to be the twisting, that's going to be the cupping, that's going to be the leaf strapping. So again, this is a kind of a diagram of our cell where two cell walls join together. And what we're seeing here is on the left-hand side of that little box that's kind of popped out is our normal cell wall, how its components are rigid and interleaved, because those give the plant its structure and its ability to support itself. And the auxins go in and interfere with the linkages involved in those cell wall components. And again, that leads to the bending and the twisting that we see. Again, it affects multiple sites. Auxins affect multiple processes and multiple sites within our plant cells. But again, cellular processes that related to cell wall development are really what we characterize the injury as. So dicamba, 2,4-D, clopyrrolid stinger, quinclorac, if you're familiar with quinclorac, which acts as an auxin on broadleaf weed species, but maybe not on grasses. It also has control of grasses. It does a different mode of action. So again, the symptomology associated with our synthetic auxins, there's the epinasty and the twisting. We see the leaf cupping, particularly with dicamba injury. That cupping is very symptomatic of dicamba contact on a plant. We see leaf strapping that's associated with several auxins. Group 9. This is the inhibition, and I'm going to say this once, of 5-enolpyruvyl shikimate 3-phosphate synthase. We call it the EPS-PS inhibitors, but you can just refer to it as glyphosate because it's the only chemical in this family. So again, the EPS-PS inhibitors, it's a foliar applied product, and it is systemic. And what it does is it interferes with the shikimic acid pathway. And this pathway also interferes with the production of amino acids, much like the ALS inhibiting herbicides, the group 2 herbicides do. But this inhibits the aromatic amino acids. So specifically the amino acids, tyrosine, tryptophan, and phenylalanine. So why is this important? Again, Similar reason as we discussed before, amino acids are building blocks of proteins. Proteins are important for the structural integrity of the cell. They're also components of enzymes that carry out the activities that are encoded in plant genes. So again, glyphosate is interfering with amino acid production and the ability to form these amino acid chains that are our proteins. It's phenylalanine, it's tyrosine, and tryptophan, the ringed amino acids. You can see the rings right there in the chemical structures on the screen. This is another picture, again, showing right where glyphosate works in this entire shikimic pathway. And again, tryptophan, phenylalanine, tyrosine being affected. And we see that those amino acids, those protein chains aren't built. But then we also know that these amino acids do have roles in the development of other plant structures and other plant chemical compounds, phytoalexins, lignins, and indole acetic acid, IAA. Again, one active ingredient, glyphosate, but there might be probably hundreds, if not thousands, of formulated products that have this active ingredient, this chemical in it. The symptomology that we see with glyphosate, we tend to see chlorosis, and usually the chlorosis starting at the base of the leaf on the youngest tissue because it's translocated to the meristematic regions. And we also see witch's broomings. One of the symptoms of glyphosate injury, and this is on apples on the left picture, is a shortening of the internodes. And when we have the shortening of the internodes, we have a lot of leaves starting to develop in bunches, very close to each other. We don't have that internode expansion, and we have these structures that are called witch's brooms developing. The next is the group 15, which is the long chain fatty acid inhibitors. These are pre-emergent herbicides that are used to control annual grasses and some small seeded broadleaf plants. They're not readily translocated. We're not as familiar with their activity within the cell, but we know that they inhibit very long chain fatty acid biosynthesis, possibly by interfering with elongase enzyme activities. And again, these very long chain fatty acids are components of cell membranes, and they're also precursors for components of the plant cuticle. There's chemicals that are part of this class that you're probably well aware of, which is dual magnum, warrant or harness, zidua. Again, dual magnum, pretty common. We use it in a lot of specialty crops here in New York. Injury that's associated with the very long chain fatty acids inhibitors are stunting and leaf distortions. Often with the leaves, you can tend to see kind of like a puckering or a crinkling of the leaves. I have seen where sometimes these chemistries get down into the whorl of plants. If they get applied over the top of a plant, they can get down into the whorl, into the growing point, and then cause distortions directly to the growing point. The group 9, this is going to be the cellulose synthesis inhibitors. And these herbicides affect cell wall biosynthesis in susceptible plants. And this inhibits cell division. So cellulose is a structural carbohydrate. It's used for protection, and it's used for structure. And the cellulose microfibrils are the structures here that are the yellow tubes. And if we can't build these cellulose microfibrils, we don't successfully build our cell walls, which means, again, we're affecting growth and development. If we can't build cell walls, we can't grow. We don't have that structural components needed for development. So this is going to have some chemicals that you may be familiar with. Casseron, if you're working in tree systems. Gallery, isoxaben, we don't have that registered in New York, but it is registered in our surrounding states. And Allion, a recent active ingredient that has been released into our, and recent relatively, our fruit crop systems that we do use in both trees and vines. Again, it's affecting cell wall development. Some of the symptoms that we see associated with the group 29 is the inhibition of cellulosynthesis are stubby roots. And you can see a picture there. It's on ground ivy, those stubby roots developing. We do see crinkled leaves and stunting of plants and plant structures. Interestingly enough, if you look at the Allion label, this is Allion damage to grapes. Allion has a very strict label for use in grapes with respect to soil type and the age of the plants. These are 3-year-old grapes on a very sandy soil in California. And this Allion got used when it was not supposed to be used. And a year later, these grapes were gone. They had to be ripped out. And this is the damage that the Allion did to these seemingly fully grown grapevines. The interesting thing is we did this damage to the grapes with the Allion. I can tell you that Allion will not touch field bindweed. Field bindweed, also a vine. You know, you would think much wimpier than a grape. Doesn't touch field bindweed, decimated these very young grapes. So why is it important to know this information? Just to reiterate what Thierry said, improved efficacy by having better knowledge or more knowledge about these herbicides and how they work. You know, it's going to help us improve our use of them and improve our weed control performance. It's going to help us understand how herbicides act within the plants and then what symptoms they're going to elicit so we can better diagnose injury. And a lot of it comes down to resistance management, because our best management practices for resistance call for diversifying sites of action. Now, we get a lot of questions, should you mix or rotate herbicides when we're talking about diversifying sites of action? So I thought I would throw this in there. There was a recent study looking at glyphosate-resistant water hemp. This came out of the University of Illinois. And what they found is if they were tank mixing multiple herbicides for application, they were less likely to have resistance. It didn't completely reduce resistance, but statistically, growers were less likely to have resistance than if you were rotating your herbicides, applying one and then switching to a different mode of action and then switching to a different mode of action. If you were tank mixing, you had the potential for less resistance. But it only works if each component of the tank mixture is effective against the target species. So we talked about the specificity of these herbicides. If only one is effective and the other isn't, you haven't really effectively mixed your products. It's not an end-all solution, because I'm going to tell you, Australia actually suggests rotating tank mixtures for managing herbicide resistance. So just don't mix, don't just rotate mixes where you can. And then of course, think about non-chemical strategies. And I do want to bring this up, this next thing, when we're talking about herbicide resistance. I just talked about those products that inhibit growth and development of the plants. All of the arrows in this picture right here, in this image right here, all of these herbicides were part of categories that I just talked about. So the only three that would have fell in the category that Thierry talked about was atrazine, paraquat, and simazine. Otherwise, we have the ACCase inhibitors, we have glyphosate, we have the ALS inhibitors, and we have 2,4-D, which is a synthetic auxin. So we have a lot of herbicide resistance, particularly to these herbicides that are associated with growth and development. So I'm just going to use that to, again, understand these herbicide sites of action, understand how they're different for managing resistance, because resistance is our #1 problem that we're facing now. Now, Thierry is going to jump to the next and last topic, which is off-target movement. So we talked about herbicides don't always work the way we want them to. Well, herbicides don't always stay where we want them to. And they can move away from an intended plant, and that is going to have repercussions for weed control, for damage to desirable crop species and to growers' economic bottom lines, as well as the environment.
All right. Thanks, Lynn. you made the introduction for my talk a little bit easier. So let's look if I can put it in presentation mode. And you should see it right now. Is it okay? Okay.
You're good.
Thank you, Lynn. So yeah, I mean, as Lynn mentioned, I mean, the last topic that Lynn and I wanted to discuss with you today was really discussing off-target movement, essentially because it's a topic, you know, I had a grad student working here with me over the last 3 years, working on off-target movement of dicamba on vegetable crops. And it's a topic for which in the Northeast region, it has not been a prominent topic over the last few years. But if you look at what's going on in the Midwest, what happened the last 2-3 years in the Midwest with dicamba crop, I think it's really important to really understand what's going on with off-target movement, what are some of the concepts that we are discussing, on how to prevent it as well, because we are not immune to this kind of problem in the Northeast region. So we'll be discussing herbicide retention in the environment. We'll be looking at how herbicide is lost over time. So it can be through degradation of movement. We'll focus on herbicide off-target movement, and we'll be finally discussing preventing off-target movement as well. So when you apply an herbicide, there is only a minimal fraction of this herbicide that will really be effective on the weeds. Actually, in most situations, most of the herbicide is reaching the soil or reaching, staying in that atmosphere for a little bit of time or things like that. But only a small fraction is actually really directed targeting the weed that you want to control. So there is many things happening. It can be taken up by the plant. With different type of herbicide, you can have volatilization, meaning that the herbicide is vaporizing, getting from a liquid formulation to a gas. You can have photochemical decomposition with some chemical family of herbicide. You can have runoff and erosion with most of them. You can have chemical breakdown depending on soil condition, especially for triazine herbicide. Most herbicide will be degraded at some point by microbes, by soil microorganisms. Sorry. You can also have leaching of the herbicide deeper into the ground, or in some situations, unfortunately, reaching the water table. On the last thing is really the interaction between the soil on the herbicide. So that's what we'll be discussing right now. So herbicide soil retention, there's two concepts that really are important to understand on what is driving these concepts. One is adsorption. I didn't say absorption, I said adsorption, which is really the retention of herbicide compounds onto the soil particle through chemical interaction, so through chemical bonding. So adsorption of this herbicide on this soil particle will determine how long herbicide will persist in the environment and potentially be available for being taken up by crops over time. So you have adsorption, but at the same time, you may have another process, which is desorption, which is basically the reversal of adsorption. You may have herbicides that can be adsorbed for some period of time on soil particle. And if soil conditions are changing, this herbicide will move back into solution, into the water solution, into the soil, and at this time may be available for taken up by crops. And that's explain what, that's one of the concepts which is explaining carryover. You have herbicide that is adsorbed to soil particle, and at some time in the season, you may have more rain, the herbicide will be back into the soil solution, into water in the soil, and be taken up by the following crops. So the retention will depend on different soil parameters. Of course, soil texture, clay soil have more capacity for absorbing herbicides than sandy soil. That will depend on organic matter content. The higher the organic matter content, the more capacity for the herbicide to be absorbed by the soil, depending on the cation exchange capacity, depending on the pH, because the pH will change the binding capacity of each herbicide by polarizing the herbicide molecule, and it will also depend on water holding capacity. It will depend on herbicide chemical characteristic. I mentioned polarity, but yes, some herbicide may be more prone to soil absorption than some other herbicide. It will depend on microbial activity. The higher the microbial activity, the faster the herbicide will be degraded in the soil. And of course, it will depend on environmental conditions. So water, humidity, temperature are all factors that will play a role in adsorption or desorption of the herbicide in the soil. So herbicide degradation, that's a different set of degradation process, but we can have photochemical degradation. So some herbicide can be really quickly be breaking down under UV light. So that's the case with some dinitroanilin herbicide. You know that, I mean, for example, trifluralin, which is a dinitroanilin herbicide, need to be buried into the soil relatively quickly after application, mostly because there is a risk of photodegradation of the herbicide molecule. And when the herbicide molecule is photodegraded, basically you're getting another compound, which may not be an herbicide anymore. The degradation, speed of degradation will depend also on microbial activity. It will depend on chemistry of the soil. So it will depend on the capacity of the soil to hydrolyze or create a redox reaction with these molecules on changing the conformation or the type of herbicide molecule. And it will depend also on metabolism with some herbicide. I mean, the plants, some herbicide are not suffering from herbicide injury, mostly because they have this metabolic capacity for breaking down the molecule of the herbicide molecule relatively rapidly. So herbicide off-target movement, that's another fate of herbicide once you applied it. So that's really when the herbicide is moving from the intended plant or site where you want the herbicide to be. So that's usually some of the consequences of herbicide moving off target is really that you're reducing your weed control because you don't have enough product where it was supposed to be. You may have a lot of unwanted damage to neighboring vegetation. It's a waste of money and time. It creates some environmental concern as well as some illegal pesticide residues. You may have herbicide drifting to a neighboring crop on which herbicide is not labeled. And if you're harvesting the crops, you may detect residues of the herbicide, even if you did not apply it. If your neighbor applied an herbicide that drifted to your field, you may end up with residue of herbicide that is not specifically labeled for this crop. And ultimately, yes, it can have huge legal and financial repercussions. So that's really a big concern that we have with off-target movement of herbicide. So we'll mostly focus on three different types of off-target movement today. We'll focus on carryover, we'll focus on vapor drift, which is chemical volatilization. We'll focus on particle drift, which is really the movement of herbicide spray into the atmosphere. But we have also other type of off-target movement. You may have movement of soil that has been contaminated with herbicide to another location, and that can create problems. You can have leaching of the herbicide deeper in the soil. You can have misapplication. You can have tank contamination that can create off-target movement as well. So herbicide carryover, we'll start with this one. So that's really the capacity of an herbicide to persist into the soil and cause potential damage to the following crop. So the potential for herbicide to carry over and cause injury to the following crops will depend on two factors essentially. One is the concentration of available herbicide remaining in the soil at the time of rotational crop planting. So again, that will depend on herbicide characteristics, soil characteristics, on environmental characteristics, essentially the weather. On the second parameter that you have to take into account is that different rotational crop may have different susceptibility to a specific herbicide. So I will show you a picture, actually, this one right here. This picture, it's showing reflex herbicide. I mentioned that before in my previous talk for Mesafen. We know, for example, that cabbage are extremely sensitive to reflex carryover, as demonstrated in this picture. I mean, you apply reflex 200 days before transplanting this cabbage, and you still have, you have a huge loss of cabbage right here on these plots. If you are planting different crops, if you are planting maybe pumpkin or squash, I think the rotational interval is just 4 months. So if you had planted a different crop, you will not see this kind of injury. So it's really crop specific. But basically, when you're looking at the herbicide carryover, if you have high clay or organic matter content, that will cause more herbicide binding to soil particle. And actually, because you have more soil binding of this herbicide, you will have less activity for microbial degradation. So microbial degradation may be slowed because you have this herbicide attached to the soil particle. And basically, since at some point in time, they can be getting back into the soil, into the water in the soil, that's a potential concern. Soil pH will affect herbicide, chemical, and microbial degradation. So microbial degradations tend to be more active around pH 7 when we have neutral soil. So if you have soil, very acidic soil like what we have here in the Pine Barrens, we know that we don't have a lot of microbial degradation of the herbicide, and that may be a concern with some residual herbicide that we are applying to this on the soil. And something important to keep in mind too, and that may explain why we have sometimes damage in spring with carryover, is that when you have cold soil, early spring, the soil is relatively cold, cold soil will decrease microbial activity, and that will slow down the process of degrading the herbicide. And when you have cold soil in spring, that's maybe sometimes a perfect condition for really getting some carryover injury early season. So the other point too is that warm and moist condition will increase microbial activity and herbicide degradation. But what is really important right here is that warm and moist, if you have a drought, if you have really, if you don't have enough water in the soil, that will slow down the activity of these microbes. They will slow down the degradation of this herbicide. And that's what you see on the curve on the right of this slide. Clearly, if you have a wet year, you can get, in some situation, you can get herbicide degradation up to the level which is safe for rotating to the next crop. You can get that in 20 weeks. If you have a dry year, it will take 3 times more to reach this threshold where you can have a level of herbicide low enough for planting the following crop. So that's something important to consider. And it's really something, if you're looking at what's going on these days, Lynn and I were looking at, a message that was posted recently regarding Illinois, regarding the Midwest. They got some drought issue last year, really dry year. And really they're concerned that this year, because of what happened in 2021, because it was a dry year in 2021, they are concerned that they may start to see some herbicide injury, an increase in herbicide injury from carryover because of condition that we had last year. So what are some of the strategies that you can adopt for reducing the risk of herbicide carryover? Well, the first thing is really, I mentioned it in my previous talk, but really look at the product label, follow these recommendations that are attached to the label regarding the rotational restriction plant back interval. Make sure that, yes, you're following that because it's really important. Ensure that seedling stress are minimized. Usually carryover is, carryover issues are more important on young seedlings and established seedlings. So everything that you can have at the time of establishment, of crop establishment for reducing stress, fertilization, moisture, fungicide, these kind of things that will reduce stress on the seedlings will also allow seedlings to survive carryover better and to have eventually better metabolic activity. If needed, you can change the crop that you're planning. If you're not sure about potential carryover injury, you can delay the planting. There is discussion that tillage may eventually be improving, but I didn't see any strong evidence that may be the case. The last thing that sometimes we don't do enough is really try to do a kind of small bioassay, a homemade bioassay. Take soil from the field where you want to plant your crop, plant something which is relatively sensitive to herbicide. In that case, in this picture, it was alfalfa. on look at, compared to a piece of soil that has not been treated, on look at both after just a few days, looking at how many seedlings have emerged in comparing a soil that has been treated with herbicides the previous year on a soil that has not been treated, coming from the same area. And that can be a good indicator of what is the potential for carryover damage. The other thing that I wanted to discuss is really herbicide drift, which is the movement of herbicide droplets or dust, as I showed you in the previous presentation, through air at the time of application or soon after application to any other site than the intended area. On these two different types of herbicide drift, there is one we are calling vapor drift, which is the movement of an herbicide as a gas or a vapor during or immediately after application. And that's the result of volatilization of the herbicide. And the second one is particle drift. It's basically these tiny droplets that are making your spray solution are moving away from where you want them to be. They're moving away and they're traveling into the air. So vapor drift, the first one, it will mostly depend on herbicide vapor pressure, which is really a chemical characteristic of each herbicide, and it will depend on formulation of the herbicide. And for example, some herbicides that are really well known for vapor drift, 2,4-D, dicamba, clomazone are herbicides that are really well known for volatilizing relatively quickly. If you're looking at formulation, on the higher the vapor pressure, the higher the risk of volatilization. So if you're looking, for example, at 2,4-D formulation, if you're looking at an amide or acid formulation, you will have a lower vapor drift with this kind of formulation than if you are using an ester formulation of 2,4-D. So actually, yes, we know that herbicide such as 2,4-D can drift, volatilize relatively easily, but the formulation is really something that can mitigate this volatilization. And actually, these days, for specialty crop, we are getting some formulation like 2,4-D choline, that's a new 2,4-D formulation by Corteva, which tends to be really low volatility, and that can eventually reduce the risk of potential of target movement. Particle drift, and that's a nice visual where you can really see that with the wind, you can see the cloud of tiny droplets moving away from the sprayer swath. So that's a nice visual right here. But really, the spray droplet size is really one fundamental parameter that is explaining particle drift. So the smaller the droplets, the greater chance that this droplet will drift to a greater distance. So usually droplet size is measured in micrometers, which is 1/25,000 of an inch, really small. To give you a comparison, a pencil lead is around 2,000 micrometers, when a human hair is around 100 micrometers, and that's the size of some of the droplets that we are spraying. So the nozzle, usually when you're applying your herbicide with a sprayer, the nozzle will produce a range of different droplet size. You will not just have a single droplet size. You will have, depending on the nozzle, you will have a range of droplet size. And usually that is described by the volume median diameter of the droplet, which is the median droplet size of a volume of spray from a nozzle. So basically, if you have in a nozzle, can spray from 50 to 200 micrometers, but half of the nozzle will be below 100 micrometers and the other half will be above 100 micrometers. So that will be the volume median diameter that we are talking here. So if you're looking at this graph, basically, yes, the bigger the droplets, the bigger the micrometers, the least they will travel over time. Actually, if you're looking at the 200 micrometers, it's not drifting far away. If you're looking at the 150 micrometers, that's the one which is drifting further away. On this one, it's drifting at some point in the atmosphere, and depending on atmosphere characteristic, especially humidity, these droplets may start disappearing by vaporizing, but you're moving from spray drift to vapor drift, basically. But basically, yes, these droplets, as physical droplets, can move over a big distance. If you start getting smaller droplets, they may stay in the atmosphere relatively long, but they can vaporize relatively quickly as well. So wind speed is something to consider as well. So when you have a 2 times increase in wind speed, that will result in 700% increase in drift, 90 feet downwind of the sprayer. So really wind speed is something to consider because that can significantly increase the risk of target movement. Usually we tend to consider that ideal wind speed for spraying herbicide is between 3 and 10 miles per hour. I didn't say 0, I already put 3 to 10. And I will explain why why 0 is not a good idea. That's why we don't, 0 is dead calm, and we don't want to spray during dead calm conditions. And the reason is that when you have dead calm condition, that may be because you have temperature inversion. On temperature inversion, it occurs when warm, light air rise upward into the atmosphere, and you have heavy, cool air that will settle near the ground and preventing the mixing of air layers. So usually it's the reverse. It's really the warm air closer to the ground will start moving upward, on the cold air is located above, higher in the altitude. Now, when you have temperature inversion, actually the cool air is really located close to the ground, and the warm air is higher in altitude. So when you have this kind of temperature inversion, basically the small droplets will remain suspended in air, and they will move to great distance laterally, horizontally, for as long as the inversion is lasting. So usually temperature inversion will begin to set up around the time of sunset. So if you have no clouds or wind to mix the air, that's perfect condition for getting a temperature inversion. So that's a nice picture where you can really see that the top picture, you have vertical mixing of the air. So you can see that the smoke is mixing, rising in the air. When you see the bottom picture where you have where the smoke is really staying as layer close to the ground, that's condition in which we have temperature inversion. So also cold air drainage can influence movement of very small herbicide droplets. So especially because cool air can drain into lower areas during the night. So because cool air is denser, it will move to areas at lower elevation, and that can be a condition that will create a temperature inversion as well. So some indicators of temperature inversion, when you have clear night and no clouds, that's perfect condition for temperature inversion. Wind below 3 miles per hour, you may have temperature inversion. If you see dew or frost, that's condition during which you may have a temperature inversion. Horizontal smoke patterns, ground fog in low-lying areas are indicative of temperature inversion. And the last one, and actually I learned about this when I was preparing the presentation, when you can hear distant sounds, then when you can hear them really well, that may be indicative of temperature inversion as well. So look at this condition, because otherwise you really don't have any way to know the temperature inversion. You will not measure the temperature 60 feet above the crop. You will not do that. So look at this kind of visual indication that are conditions during which you don't want to apply an herbicide. So relative humidity and temperature are also important to consider regarding drift, physical drift. So if you have low relative humidity and high temperature, basically the herbicide, the droplets will be reduced in size because you will have some water evaporation. On the more the herbicide, the more the droplet is reduced in size, the further it can travel with the wind. If you have high relative humidity, low temperature, you don't have so much evaporation from the spray droplets, and the droplets will not travel on a longer distance. Spray pressure is also important to consider. So the higher the pressure, the smaller the droplets. So the relationship between the two, between pressure and flow rate, it's not a linear relationship. Actually, if you're multiplying the pressure by 4, you will have an increase by 2 times of the flow rate. On increasing that, will reduce the droplet size as well. So selecting the proper nozzle is also important to consider. If you have, it will depend on the type of herbicide that you're spraying. If you're spraying systemic herbicide, that can move into the plants from the area where they're reaching the plant to a different area into the plant, you can consider getting coarse droplet because the herbicide will be there. It will move, then when it's absorbed by the plant, it can move into the plant. Now, if you're looking at contact herbicide like bromoxone, you need to maximize the surface of the weed that will receive herbicide because these herbicides are not moving into the plant. So in this condition, you may be using a medium-sized nozzle for this type of herbicide. So is there a pesticide contact or systemic? We just discussed that. So if you have drift concern as well, so if you're looking at the label, some label will specifically indicate that you need to use some specific nozzle because there is potential drift concern with this type of herbicide. So looking at the label is always a good idea. Oops. Yeah, here we go. So if you're looking at selecting the proper nozzle, so nozzles are always color-coded. Red nozzle correspond to the very fine spray droplets. And usually with herbicide, we really recommend using as much as possible, either white, green, or blue, eventually yellow. But in my program on discussing with growers, we really try to say to growers, do not use red or orange because it's a recipe for getting some off-target movement. So selecting the, I mentioned it before, but selecting the proper nozzle will depend on the type of herbicide. So if you have contact foliar herbicide, medium-sized nozzle will be better. If you have soil-applied herbicide, you can use coarse or very coarse nozzle for this type of herbicides. Boom height is also important to consider. So the higher the boom, the greater the drift potential. A 2 times increase in height will result in 350% increase in drift, 95 downwind of the sprayer. So that's something. Keep the boom as close as possible from the crop to reduce potential of target movement. If you're looking at the TJET catalog, actually, really, they are providing you with indication on what is the optimal height that you need to follow when depending on the type of nozzle that you're using. So it's relatively, I would say for most of the nozzle, we try to keep it between 16 and 18 inches above the crop. But that will depend on, for some of the nozzle, you can go up to 20, 24 inches. Follow the label. Again, I put a picture of Callisto label, but you have restriction on the label. So make sure that you're reading the label carefully because that can provide you information regarding the type of nozzle that you need to use or regarding condition that during which you can apply the herbicide for. So for example, for Callisto, they clearly said that you cannot apply this herbicide when the wind speed is exceeding 10 miles per hour. And you cannot apply it during temperature inversion as well. So other tips, other tricks is sometimes you can use shielded sprayer, especially in vegetable crops when we're using post-emergence herbicide. I know that we try to do our best to use shielded sprayer because you can get off-target movement with any kind of herbicide. And since vegetable crops are extremely sensitive to herbicide injury from drift, we try to use shielded sprayer for that. Use low volatility formulation of 2,4-D or dicamba if you have to use this herbicide. Do not forget to eventually add drift reducing adjuvants to increase the number of large droplets when possible. Lower the boom height, we discussed that. Maintain buffers to sensitive crops. That's something important to look through on the label as well, is that the label may have indication that you need to maintain a certain distance from when you're spraying from a neighboring crop that may be sensitive to this herbicide. So pay attention to this buffer zone. Don't spray during high humidity. I know, high humidity may be causing off-target movement. Some herbicide, you really don't have the choice because I was mentioning glufosinate before. We prefer to spray glufosinate when we have high humidity, but you need to be extremely careful when you do that it's not drifting to neighboring crops. If you're adding the humidity, the percentage of humidity on the air temperature, if you're reaching 125, you do not spray. Spray when, it's a rule of thumb, but spray when the condition, when humidity and air temperature, when you're adding both together are less than 125. monitor wind and other environmental factors, properly clean the sprayer when you're done, because one other way of getting off-target movement is really to get tongue contamination. I didn't discuss it, but really making sure, especially with 2,4-D or dicamba herbicide that are really acting at low concentration, make sure you really do a proper cleaning of your spray tank, because you don't need a lot with this herbicide. I mean, Lynn and I conducted studies over the last 2 years. Depending on the crop, you may just need 1/5,000 of the labor rate to see damage on the crop. I put a picture of dicamba injury on eggplant. On actually, this one was we observed this kind of damage at 1/1,000 of the labor rate. We start to see some injury with dicamba. So cleaning up the tank and making sure that you don't have herbicide residue in the tank is really important, especially with some of these herbicides that are really prone to drift. And with that, that will conclude my presentation on off-target movement. I don't know if we have questions or if we have time for questions, but yeah, I mean, if we have time, I will take any question.
Thanks, Thierry. I know Gemma is probably going to bring up the CCA credit scan, the QR code for CCA scans. And in the chat, she has put the sign out for the DEC. I want to thank Thierry for presenting with me today. I also want to thank Thierry for, again, all the work we're doing together. If you were at the snapping session yesterday, I actually discussed our dicamba injury results and how dicamba injury can induce stress in snappings, which can lead to yield loss. Thierry and I can stay on for a couple minutes, so if you do have questions, you can put them in the chat or you can ask them. If not, thank you so much for attending. And I will be working with Thierry to write up a summary of the very general information, not necessarily all the details, for inclusion in the abstracts for the meeting. Also, if you subscribe to the Veg Edge newsletter, Thierry and I put together an article that came out a few weeks ago, maybe a month ago, on the herbicide shortages. We are currently working on an article about drift, carryover, volatility, tank contamination, leaching, and runoff impacts. Hopefully we'll have that for April for the Veg Edge newsletter, some of our other Cornell newsletters, as well as Rutgers website. And Thierry and I are just going to be working together on a lot of extension outreach such as that. And Justin put in the chat about whether work has been done on soil surfactants and the movement of pre-emergent herbicides. And yes, soil surfactants are out there. I personally have not worked with it, but I do have a soil surfactant that I got from a company that we're going to be investigating. I don't know if Thierry has done work with soil surfactants.
I did one study last year, and it was looking at soil surfactant in cranberry. Actually, I mean, they're using soil surfactant not for herbicide, but mostly for some fungicide that are affecting cranberry roots. And we look at adding this surfactant, which is supposed to increase fungicide efficacy. We added the surfactant to one of our pre-emergence applications. Unfortunately, I mean, we used it with sulfentrazone. On actually the use of the surfactant increase cranberry damage caused by sulfentrazone. So that was a case where, I mean, I think the herbicide was maybe not appropriate because we already know that sulfentrazone, if it's not applied at the right stage, has also post-emergence activity and can potentially cause damage. But I think that it's an area where we need to get a little bit more information, essentially because we mostly recommend to use pre-emergence without surfactant. But maybe in some situation, getting a surfactant may improve pre-emergence efficacy. But my experience was limited. I think, yes, it's a field to investigate.
Yeah, again, my experience is limited. I have talked to companies about it. I'm going to be doing some work to see if we can use some soil surfactants to in sort of safer herbicides by maybe preventing movement outside of a treated zone and into like our where our crop is seeded, or to prevent herbicide movement, keeping it in that zone where the weed seeds are most germinable, and preventing it moving into where the root balls of our transplanted crops would be. Again, for me, it's exploratory at this time. We have a product in hand. We're going to test it this year. I think Christy Hochting is also hoping to do some tests in cabbage for much the same reason, with an alternate soil surfactant chemical. But also, I will say, I don't know about Thierry, but Alan Taylor, who's our seed scientist here at Cornell Agritech in Geneva, he and I are also looking at some seed treatments, not necessarily to keep herbicides from moving, but that might be able to actually act as a protectant and reduce some of the injury potential on some of our seeded crops. So I think both Thierry and I are looking at, how can we reduce damage using different opportunities, whether they're soil surfactants or seed treatments, to safen some of these chemistries in our crops.
Actually, Lynn, if you have, that's something that we can discuss that later. But if you have the contact information for the products that you may want to test this year, I may have some interest for cranberry, definitely.
Yeah, I will reach out to you, Thierry. Again, if you have questions, you can type it into the chat. I know there's 47 participants hanging on. Are we able to accept samples to do residual bioassays? No, we don't do that. I don't do that. That's a good question about the labs, though. And Mary Centrella, I don't know if she's on, that maybe she would know of labs that do testing, or you might also reach out to your local CCE specialist who might know of labs that would be able to do testing. I'm not going to do bioassay testing, but I am willing to accept your weed seed from plants that don't die for herbicide resistance screening.
Actually, Lynn, that's something that I did a few times, mostly for for making sure that when I was suspecting injury from Carryover, I took some sample from a field on look at some sensitive crop. And to me, I'm using it mostly as a way to confirm what I've seen in the field.
Yes.
But I don't know that here in New Jersey we don't. I don't think that the Rutgers Soil Lab. I'm quite sure actually they do not do that. It's not a service that they're proposing. So it's mostly up to the grower to do that to evaluate the potential for Carryover.
Lynn, I'm sorry, I missed what you were talking. What kind of testing you were talking about?
Yeah, if you know of labs that perform soil testing for residual herbicides or bioassays, do you know of any labs?
Yes. Well, I'm not sure if it's changed since I was there, but I know within entomology, if you talk to Scott McGart, who's a bee researcher, he used to have a lab technician that would do all sorts of samples, and you could pay to get those samples ran. I'll look into it for a second, try to put something in the chat here and see if it's still operating. Because I know his technician left, but they might still have someone in that position.
Yeah, so that's great. Again, I don't do it mostly because I'm also trying to, for our greenhouses, avoid bringing in external soil that could have pests or pathogens. We'll bring obviously seed in that we've thoroughly cleaned, but I do try to avoid bringing soil into our greenhouses just from a phytosanitary standpoint.
I just put a link in the chat too, so it looks dead. I believe this site works pretty well. You can actually put in the chemical you're looking to get a sample for, and they might be able to help you out.
I don't see a link in the chat, Dan.
It is there now.
Yeah.
I was just typing it in, so yeah.
So again, if you've got any questions, we got 30 people on, feel free to throw them in the chat. Let us know. Again, thank you to our sponsors. Thank you to Gemma for running this session behind the scenes. Thank you very much to all of the CCE personnel who are involved in developing the expo and then bringing the expo from a in-person to a virtual session in very short notice, considering the situation at the time. Hopefully we will be back next year. Thierry and I are already discussing about possibilities for future talks, which would review the different mechanisms associated with herbicide resistance, the current state of herbicide resistance and all the resistance mechanisms, from target site mechanisms to non-target site mechanisms. Thierry and I have also talked about the possibility of, in the summer, if we're able to hold a session here at Geneva Agritech, of having a hands-on herbicide symptomology field day and field site, or at least Thierry and I have talked about it, and I will just badger him in further and rope him in to do it so that people can actually come and see the injury firsthand. That's going to depend on a lot of things, though, and particularly where COVID goes within the next 6 months.
It's good. We already have our talk scheduled for next year. It's not so common.
If you're going to be attending tomorrow's cover crop talk, Thierry will be speaking in the cover crop session. And if you are a field crops grower and you've been following Jeff Miller's, he's been sending out blasts about different weed science and weed control related talks. On the 15th or the 16th, I don't remember which, I'm going to be talking about our non-target or our non-chemical weed control technology for resistance management. So we'll be talking about harvest weed seed destroyers. We'll be talking about electric weeders and other robotic implements, the kind of the future of weed control. So you can register for, I believe, for free for that.
I just wanted to ask that I did find that link. So I put that in the chat there for the pesticide analysis facility at Cornell. It is a little bit expensive per sample.
Yeah, both Dan and Mary have put links in there that could be valuable for people interested in this. All right, well, if there is no further discussion, I'm gonna close this down. Again, thank you so much to Thierry, thank you, Gemma. Thank you, Dan and Mary, for contributions.
You're welcome, Lynn, and thanks everyone for inviting me to come today. Great job for moving that from in-person to online. It's really great.
Thierry, you're an honorary Cornell person now.
Thanks, Steve. I still have to come to visit Lynn in July, like we discussed.
Very good. We'll see you tomorrow then.
See you.
Okay.
All right, thank you
so much.
This has been a production of Cornell University, on the web at cornell.edu.